first, I'll just start off with our introduction. So this will take maybe 4 minutes, about 3 or 4 minutes. But welcome everyone. I'm Robert Driver again. I'm a postdoc at Duke University, and welcome to the AOS Student Research Grant Seminar Series. And first, like I do each week, I'll just introduce the AOS Student Research Grant, which is a yearly competition, which is very competitive, that the AOS hosts, where research money is given out to early career researchers for projects in ornithology. And it's a challenging call to be successful, because you have to advance a project in the field of ornithology while still remaining feasible within maybe a graduate dissertation to still be within the AOS's student research grant budget. So our seminar series here, the AOS Student Research Grant Seminar Series, celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing this work to the AOS members. And each week we're showcasing the research and research plans of a 2023 or 2022 AOS Student Research Grant winner in a seminar-style format. And so we have one seminar each week for 15 weeks. I think this is week 4, maybe. And I want to thank everyone for being here. Thank the AOS, thank the AOS Student Affairs Committee, and thank Wiliam for being our speaker today. And so before we get to Wiliam's research, I just want to mention to put your microphones on mute if you haven't already. If you have any questions, there will be time at the end of Wiliam's talk for a Q&A session, and you can type your question in the Q&A box in Zoom feature, the Q&A Zoom feature, although I believe that the chat is now enabled for attendees, so you could also potentially type your question in the chat, or you can raise your hand and unmute yourself and ask your question by speaking it. So I wanted to remind everyone that this is an AOS sponsored event, and that all participants are required to follow the AOS virtual meeting event code of professional conduct. And I'll post a link to the AOS virtual meeting event code of professional conduct in the chat. And please be aware that we are recording this talk. I'll also post a link to a post-seminar evaluation form that you can fill out to let us know how to make these seminars better. We're going to have these seminars weekly, and we're always looking to improve. And I'll also send an email to contact the Student Affairs Committee if you have any questions. Okay, so now for this week's speaker. Wiliam Peralta is currently a Ph.D. candidate in Conservation and Natural Resources Management at the National Polytechnic Institute in Oaxaca. This is CIIDIR-OAXACA. That's the correct way to say the acronym? Okay, great. CIIDIR-OAXACA, okay. Osaka. Okay, William's academic career began with a BS in biology from the Autonomous University in Campeche, working with Jesus Vargas Soriano. And following graduation, Wiliam gained a wealth of experience working under different research projects, monitoring and inventorying birds in Mexico. And after these internships, Wiliam began his master's degree in Conservation and Natural Resources Management at National Polytechnic Institute with Dr. Jose Roberto Sosa Lopez. And this is where he began working with his current student study system with these wrens. His master's research was published in no other than the AUK. And in 2020, Wiliam stayed on in the lab of Dr. Lopez to begin his PhD. Wiliam's research is published in Environmental Science and Pollution Research, Animal Behavior, and Ornithologia Colombiana. In addition to the AOS Student Research Grant, Wiliam has also won the Developing Nations Research Award from the Animal Behavior Society, whose logo is on this slide right here. So with that, I will turn it to Wiliam for his talk, Asymmetrical Responses to Multimodal Signals in Two Neotropical Sister Species of Songbird.
Okay, thank you so much, Robert, and thank you for the invitation to this series of seminars. First of all, hi and good afternoon to everyone. As Robert said, I'm Wiliam. And today I'm excited to share with you guys one of the chapters of my doctoral thesis. So in this chapter, my advisor and I explored how closely related bird species use multimodal signals for species recognition. In this chapter, we aim to understand how these signals, such as vocalizations and plumage, work together to help birds distinguish between conspecific and other species. By examining these communication strategies, we gain insights into the evolutionary processes that shape species interactions and help maintain species boundaries. I hope you find this topic as fascinating as we did while we were researching it. So, with that, I'm going to start talking about the um, the topic. So, um, first, I'd like to start by mentioning that animals use a wide array of signals across different modalities to communicate with members of their own and other species. These signals modalities can include visual signals, chemical signals, acoustic signals, and tactile signals. So, variation in the structure of these signals may influence crucial behaviors such as species recognitions and mate choice, playing roles in evolutionary processes that lead to formation of premating barriers and that maintain reproductive isolation. However, animals can also integrate multiple signals from different modalities into a single display when communicating with others. Receivers may access the signals simultaneously, and this process is known as multimodal signaling. Multimodal signaling is thought to enhance communication by increasing the amount of information conveyed. These can facilitate species recognition, social interactions, and mate choice in diverse ecological contexts. So with that, I want to say that multimodal signaling has been documented across a variety of taxa in nature. For instance, frogs may use a combination of acoustic and visual signals to attract mates. Similarly, fishes may use a combination of visual and chemical signals. Um, sorry, I saw a question in the chat. Similarly, fishes may use a combination of visual and chemical signals for mate attraction. Birds can combine. Sorry, is there any problem with my audio?
Yeah, William, we're just talking about how to fix your Wren songs, but I think you can just continue along with the presentation and we'll check on that when that slide comes up.
Okay.
Okay.
Um, yes, I was talking about fishes, and fishes may use a combination of visual and chemical signals for also for mate attraction. Birds can combine visual and acoustic signals as well. And in some bird species, acoustic, visual, and even vibratory signals can be integrated into a single display. So, in situations of multimodal signaling, each component of the signal may convey different or similar functional information. For example, there can be redundant signals where different components provide functionally similar information, thus reinforcing the message. So there also can be non-redundant signals where different components provide functionally different information, complementing each other, and there can be interaction between signals, where one component modulates the perception of the others, enhancing or altering the overall message. Hence, stronger responses to specific components, of a multimodal signals can indicate that those components may be under strong selection pressures. For example, it has been documented that in some bird species, plumage played stronger roles during species recognition, and in other species, song is more important than plumage for species recognition. So then, why studying multimodal signals. It is important to understand how males and females use multimodal signals during the species recognition process. Moreover, it is essential to elucidate how animals respond to divergent multimodal signals to understand their contribution to reproductive isolation and therefore to the maintenance of biological diversity. So thus, and this study, we investigated the role of multimodal signals in species recognition and the maintenance of reproductive isolation in two neotropical wren sister species, the rhesus gnat wren, Campylorhynchus humilis, and the Veracruz wren, Campylorhynchus rufinuca, in a simulated secondary contact scenario. We did this by examining how these species use divergent multimodal signals for species recognition, and to achieve this, we addressed three key questions. The first one, do species discriminate between con and heterospecific multimodal signals? The second, do sexes respond similarly to female or male con and heterospecific multimodal signals? And the third one, do species and sexes attend more to plumage or song during specific recognition? So, sorry, to answer these questions, we conducted reciprocal play experiments using vocalizations from both species, combining them with taxidermic mounts. But why we did choose this species for our study? Because they have an interesting evolutionary history. Both species inhabit tropical dry forests in Mexico. Rusnap wrens range from Colima to Chiapas, while Veracruz wrens have a small isolated population in central Veracruz. Both sexes sing, with males producing a more complex repertoire of solo songs, while females produce simple trill-like songs. They combine their songs to produce duets. Genetic evidence suggests the relative recent divergence, and both species were recently recognized as different species by the American Ornithological Society. Although their songs sound very similar to the human ear, statistical analysis revealed significant differences. Additionally, their plumage patterns also show some degree of divergence. However, the role of vocalizations and plumage in conspecific discrimination in these two species remains uncertain. I hope you can hear the songs of the species, so I'm gonna try to put them.
Yeah, William, we're getting some suggestions here that there is a share computer audio uh, feature in PowerPoint. Uh, and if you're able to find that to turn that on.
Okay,
Oh,
share.
I'm sorry. In Zoom, Zoom has a share computer audio option. Uh, or is it in PowerPoint?
Select microphone, select a speaker, audio settings. Yes. It can't, it can't find this.
Okay.
Filter, video settings, and no.
Yeah, it might not be easy to find. So I was hoping that would be an easy, quick solution.
Windows system. Okay,
Okay.
so let's just try if you can.
Yeah, I think we'll just go forward if it's. Oh,
Did you hear them?
no.
Um, sorry. Yeah, I cannot find that option. in
So Robert, we're getting a message that, yeah, Robert and William, we're getting a message that if you stop sharing and then you share again with audio, that might solve the problem.
Okay, let me try that.
Okay.
So I'm gonna stop share, and then I think share sound. Ah, okay. I think I found it.
That's great. I have to turn down my headphones now. Yeah, perfect. Thanks for everybody's patience. It's great, William.
So
Yeah.
sorry and thank you. So that was the song of the female of the roost not wrens. So let's hear the male of the roost not wren. And I'm gonna put the song of the female of the Berkeley wren and the song of the males of Berkeley wren. Yeah, that was all. So why did we did choose this species? So because they're very interesting. Yeah. So when I was starting doing my masters, I easily confused the songs of the both species. So so we asked how the species are using the vocalizations and plumage for species recognition on, or how they would use these signals in the case of a secondary contact scenario. So, sorry, got lost. So, to conduct the study, we hypothesized that if birds discriminate between divergent multimodal signals by using them as redundant, they would show a more aggressive responses toward treatments with both conspecific song and mount stimuli. They also would show intermediate responses toward treatments with a combination of conspecific and heterospecific song and taxidermic mount stimuli. And birds would show weakest responses towards treatments with heterospecific song and mount, as well as to the control. So in this study, we used as control signals from great kiskadis because they are sympatric with both wren species across much of their range. Therefore, they may represent a potential ecological competitor, but not sexual one. Additionally, we also predicted that if song plays a relative more important role in species recognition, birds would exhibit stronger responses to treatments involving conspecific song paired with heterospecific mount, compared to treatments involving heterospecific song paired with conspecific mounts. Moreover, if plumage plays a more significant role in species recognition, we predicted that birds would exhibit stronger responses to the treatment combining heterospecific song with conspecific mounts compared to the treatments combining conspecific song with heterospecific mounts. In general, this research was conducted using pairs of territorial birds captured and banded in Tlacotalpan in Veracruz, Mexico, and Juchitán, Oaxaca, Mexico. We applied treatments featuring different combinations of conspecific and heterospecific mounts and songs to territorial pairs and performed statistical analysis on the measured variables. For the experimental playbacks, we used solo songs from different individuals of both sexes recorded from non-local populations. And for the visual stimulus, we used one taxidermic mount per sex of each species. As a control, as I said, we used songs and mounts of great Quiscadis. All taxidermic mounts were provided by the Museo de Zoología Alfonso L. Herrera at the Universidad Nacional Autónoma de México. Each pair in the study was presented with a combination of acoustic and visual stimuli. So the combinations were conspecific song and mounts, conspecific song, heterospecific mount, heterospecific song and conspecific mount, and heterospecific song and mounts, and with the control song and mounts. The trials lasted 7 minutes, comprising 2 minutes of observation, 3 minutes of stimulus presentation, and 2 minutes of a silent period. And to record the variables, we assessed the bird's behavior during the 3-minute presentation period and the 2-minute silent period. During the trials, the observer was hidden behind the bushes approximately 20 meters from the playback, from the speaker and mounts. We recorded 11 variables, which are number of solo songs, number of duets, latency to approach to 10 meters, time spent within the 10 meters, latency to approach to 5 meters, time spent within 5 meters, the closest approach to the speaker and mount, number of paces over the speaker and mount, number of attacks to the mount, latency of attack to the mount, and time spent attacking the mount. So, using all the registered variables, we conducted principal component analysis to reduce the number of variables into orthogonal components that provided measures of aggression towards the treatments. This analysis retained 2 main components, which explained most of the variance. So the first principal component was primarily associated with vocal behaviors and physical approaches to the speaker and mount, such as the number of songs, duets, and paces over the speaker and mount. And the second principal component was linked to aggressive behaviors towards the mount, including the number of pecks, time spent attacking the mount, and latency to attack the mount. So, to analyze Wren's responses, we conducted 2 nested generalized linear mixed model, one for each principal component. In the first model, we included the terms species, sex of the stimuli, sex of the stimulus nested within sex of the territory holder, and nested within species. We also included treatments nested within sex of the presented stimulus, nested within sex of the territory holder, and nested within species. Then, in the second model, we included treatment order nested within the sex of the territory holder and nested within species. Sex of the presented stimulus nested within sex of the territory holder and nested within species. Treatments nested within sex of the presented stimulus, nested within sex of the territory holder, and nested within species. All these terms included in the models were the results of removing non-significant terms from the analysis. So what we find after all this analysis, overall, what we found is that roost-gnat wrens were more aggressive than Veracruz wrens. So roost-gnat wrens were more vocal, approached closer to the speaker, and spent more time near the speaker and the mount. We also found that responses varied based on the sex of the presented stimuli. In the case of roost-gnat wrens, both males and females were equally aggressive towards the female and male stimuli. This means that both sexes were very vocal, approached closer, and spent more time near the speaker and mount. Regarding principal component 2, we found that females respond more aggressively to treatments that came from females. regarding responses to the different combinations of treatments for PC1 when the treatments came from females. In general, both sexes responded with stronger intensities to conspecific song and mounts. Both sexes showed intermediate responses to mismatched signals, and the weakest responses were observed to heterospecific signals and the control. Responses to conspecific mounts and song differed significantly from most of the treatments, except from the heterospecific mount combined with conspecific song. And responses to the control differed significantly from most of the treatments, except from the heterospecific mount and song. When the stimuli came from males, the responses to different treatments followed similar patterns to those observed when the stimuli came from females in roost-gnat wrens. However, in this case, responses to the control did not differ significantly from those given to the conspecific mount paired with the heterospecific song. In PC2, for the roost-gnat wrens, when the stimuli came from females, only females responded with more intensity to treatments featuring conspecific signals and with the weakest intensity to the control. This suggests that females can discriminate between the control and conspecific stimuli. In PC2, when the stimuli came from males, non-significant differences were observed among treatments, which suggests that that there's no significant responses or aggression to the speaker when referring to aggressions to the mouth. Sorry. So, regarding responses of Veracruz wrens to female and male stimuli, both sexes responded with higher intensity to male stimuli. So, in the case of principal component 2, there were no significant differences in the responses to the sex of the stimuli between the sexes of the focal subjects. Regarding responses of Veracruz wrens when the stimuli came from females, only males discriminated between the treatment with the heterospecific mount and conspecific song, the conspecific mount paired with the heterospecific song, and the contrary. However, for the same principal component and when the stimuli came from males, both sexes discriminated between the control and the rest of the treatments. Non-significant differences were detected in principal component 2 for Veracruz wrens when the stimuli came from females, and nor when the stimuli came from females. So, what these findings mean? First of all, the responses observed in roost-gnat wrens matched our predictions. This suggests that song and plumage may be functioning as redundant signals. But why does this species use multimodal signals to communicate? We believe one possible explanation is that it enhances transmission effectiveness, increasing the information content or the efficacy of eliciting responses. However, we are aware that we only use one taxidermic mount per species, which may have introduced some bias into the results. So we encourage future studies to explore whether these signals are truly redundant for roost-gnat wrens, or if the presence of one signal enhances the detection or the other. This to better understand the adaptive significance of multimodal communication in this species. So, our results also suggest that roost-gnat wren's song may play a more dominant role in the recognition of males, but not in the recognition of females. However, there are contrasting findings in the literature suggesting a stronger role of plumage or similar importance for both signals during the species recognition process. While all these findings highlight the varying roles of song and plumage in species recognition, The relative importance of each signal modality may be influenced by different factors, such as habitat. For example, in denser habitats, acoustic signals may be functioning for long-range communication, while in open environments, plumage coloration may be more important for communication. In the case of roost-napped wrens, we propose that songs serve as long-range signals that elicit initial responses, while plumage may function as short-range signals, potentially adding fine-scale discrimination to the process. Thus, wrens may be using this signal in a sequential manner. Another question remains respect to our findings. Why do species respond asymmetrically to multimodal signals? So these kinds of responses are thought to arise from differences in evolutionary history, ecology, or cognitive abilities. One possible explanation for the pattern we observe is that increased population abundance may provoke an aggression toward conspecifics due to competition for resources. Alternatively, the species may have different breeding seasons, which could explain why veracruz wrens did not perceive roost-napped wrens as a threat. Another possibility is that the smaller populations of veracruz wrens may result in less variation in their communication systems due to limited gene pools and fewer environmental pressures. So, in this study, we observed that both sexes engaged in territory defense. This is a common behavior in tropical species where both sexes need to defend their territories year-round. It has been proposed that long-term monogamy may be linked to this type of behaviors, and therefore this behavior may stem from cooperative territorial defense. So, as conclusions, our study sheds light on how both sexes use multimodal signals during species recognition. In roost-napped wrens, we found that signals may act as redundant, with songs being relatively more important than plumage, but also suggesting that these signals may be used in sequential manner. Additionally, the study highlights how both sexes use signals during territory defense. And with that, I want to acknowledge and I want to thank, the authorities and landowners from the study sites who allowed me to carry out my research, to all my lab mates for their support, to CONACYT for the scholarships provided for my postgraduate studies, and to the Instituto Politécnico Nacional and the Postgraduate Secretariat for their financial support, and also to the American Ornithological Society and the Animal Behavior Society for the grants they awarded me during my postgraduate studies. And to all of you who attended my talk, thank you.
All right, thank you so much, William. A lot of Wren data to digest there for everybody. Thanks so much for sharing your work with us. And we can have the audience type any questions in the Q&A feature or the chat, or raise your hand and unmute yourself. Many ways to participate. Ron says, nice experimental design and great results, which I agree with. I just wanted to, I'll start off by asking, for the control, is it known that the wrens don't care about the kiskadee then?
So what I observed while I was performing the experiments is that they usually gave some amount of responses to the great kiskadees. So I believe this is because they great kiskadees and wrens, they usually compete for acacia trees when both species nest. So that's why I choose the kiskadees as a control, because I really wanted to know, so if there are responses to the great kiskaris, so this may be because they are ecological competitors, but not sexual. So this would give me just kind of measurement of how they may, how wrens may respond to sexual competitors. So I don't know if I responded to your question.
Yeah, I understand it was just a little bit different than what I expected based on your model figure bar graph, where control was it was highlighted in red and was like low, similar to, I believe, the heterospecific red bar. Um, so that's why I was expecting that uh, bar to be a species that, yeah. So for example, control is low and red there. So I was sort of expecting the species selected there to be one that did not cause aggression levels.
So would you repeat that for me, please? I
Yeah,
couldn't hear.
sure. Like so yeah, if you leave the graph that graph up with the red bar, which is your model, um, so you were saying you chose, I understand your explanation, your answer totally makes sense. You chose the Kiskadee because it could be a different way that the wrens are engaging in aggressive behavior, say for nest competition as opposed to some sexual territory competition. But in the model, your control bar is predicting a very low level of aggression, which would lead me to expect that the selection for that control was a bird that the wren naturally has no uh, aggressive level with.
Okay.
Does that make sense?
Uh, yeah, yeah, I couldn't understand like, but like,
Okay. Okay. I'll try. We'll move on to Farzan has a question here, but I'll repeat it. Um, why did you use these two species of birds, being the wrens, I believe, rather than another species? So how many total number of species of this bird are there? Maybe she's referring to the species complex.
oh, yeah. So I used this species because um, when I was doing my master's, I was like wondering about how they recognize each other, um, given that they are very similar. So to naive observers, naive observers can confuse um, the species, and actually, I found a new record of Veracruz wrens like registered in the near to Juchitan, where they don't really range. So I asked if they are very similar, how they tell apart. How would they tell apart each other in a potential scenario contact scenario? So that made me to choose these two species for my study. And how many total number of species of these birds are in the complex? So there are like three species that are very closely related, which is the Veracruz wren and the Rusnap wrens and the Rufus back wren. But the Rufus back wren is like more uh, it's not like very close to to the Veracruz wren and the Rusnap wren, as both species are to each other. So, so I maybe will be conducting another experiment with that species.
Uh, yeah, I just wanted to. So I'll ask in sort of a different way. Um, So what if a control was um just a mount? or and no speaker, or just a speaker and no mount. Do you think that um, that has any value?
Oh, okay. So if the so actually we performed another experiment with this same species, and we performed those experiments with only the speaker.
Mhm.
So the responses were very, very, very different. So
Hmm.
in some cases, the um the birds only approach to the speaker, and they uh like um they found that there were no um you know there were no threats to their territory or maybe to others, so they just fly away. So with this experiment, so we so this experiment allowed us to measure how they may respond in a more close way to the reality, using the mounts. So we had the opportunity to to measure this aggression in the case when there was an intruder. So I think the responses will vary a lot if there was no mount or just the mount, for example. So in this case, to the control, I think uh, the if we performed the experiment without the songs of the and only the mount of the great Kiskadis, we would have no found no responses to the control. I believe.
I see.
Mhm.
Yeah, and feel free to type any additional questions, but until I see them, I'll just try one more time because we have the time, so um, I might as well try to explain it. So, like the the in this model, and anyone else can jump in if I'm doing a terrible job or I might not be explaining this right. But in this model, the control red bar is low, right?
Yeah.
So your control is similar to heterospecific, low aggressive interactions.
Okay.
So if you wanted to if this is your prediction, I would have thought, and I'm selecting the control bird. I would have thought that the control bird selected would would produce a low aggression level in the wren, right?
Yeah. Yeah.
But the Kiskadee is not that kind of bird, right? It's a bird that does produce a high level of aggression in the wren. So I wanted to ask about that as a way that your results maybe weren't consistent with your model.
Yeah. So okay, in some cases, for example, so like in like in this one, for example. So usually the control elicited a low response, but not like the lowest as I really expected. So this means that so there's certain type of competition between species, I think. But the significant differences that were given, so that we found between the treatments with both conspecific signals and the difference between the control may give us an insight that the competition between two species is there, but not as high as when you have the two species competing for in a certain sexual way. Yeah. So my reasoning is that, but I don't yeah, I'm not sure if I convinced you with my answer.
No, I think it's good because you're actually able to say more, I think, in a certain way because of your control. was a bird that Wren is not interested in at all. That would be
Yeah,
my choice of a control if I had to design it. But if that was the case, then you could prove that there was heterospecific and within the Wren aggression that was above that level of a lack of control. But by selecting the Kiskadee, you're able to show if you see a significant difference, you're able to show that sexual aggression is above uh, what were you saying, nest site competition, for example, aggression. So
yeah,
so you're able to say a little bit more there. So I do understand if that.
yeah. And also, my this experiment was like, you know, um, designed for, you know, like proving that there would be like kind of certain interaction between the species in the sexual way. So because
I
they
see.
very closely related, so we may think that closely related species may interact if they come into contact. So in the sexual um, as sexual competition. So we wanted to really focus or experiment in that way, proving uh, maybe kind of certain sexual competition.
I understand. Yeah, makes sense. Thank you. And then Eugene says here, were the playback songs in or not in the repertoires of the focal males in each species?
Were the playback songs in or not in the repertoires of the vocal rate males? Yeah, yeah, they were. Yeah, yeah. I actually went to some place to record the bird songs, and I selected those songs in a random way to introduce them into my stimuli. Yeah.
All right, great. Thanks, everyone. I'm glad we got the songs working. I'm glad we all got to hear the songs.
Yeah, me too. Yeah.
And we'll know that for next time about audio.
Mhm.
Um, but I really want to thank William for sharing his research here with us and a lot of data that I really enjoyed seeing. And so thanks so much. And thank you everyone for being here. And we will see you guys next week then.
Thank you, Robert, and thank you all for attending to the talk. Okay.
All right, everyone. Thanks again, William. Bye, guys.
Bye.